Auzubillahiminasyaitonirrajim. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. I'm Muhammad Eko Syafiq bin Masnun, S48588 from the Faculty of Fisheries and Food Science and under the supervision of Dr. Sharifah Nur Emilia binti Sheikh Jamil Fada. And today I would like to talk about the effect of probiotic bacillus species on grouper culture in improving immune response against vibrosis. A review. For my problem statement, the first one, orange spotted grouper have slow growth performance. The second one, hybrid grouper, cause imbalance in wild ecosystem. The third one, vibrosis has been considered as one of the major diseases in Epinephrus coides. And the fourth one, the severity of vibrosis has increased. So the main objective of this case review is to study the effect of probiotic bacillus species on grouper culture in improving immune response against vibrosis. Okay, for my experimental design, grouper Epinephrus coides juvenile were purchased from a private farm in Pingtung, Taiwan, were shipped to the Department of Aquaculture, National Pingtung University of Science and Technology. The fish were acclimated indoors in a 2-ton tank with recirculating aerated seawater and fed with a controlled diet without subtilities for 2 weeks before the experiment began. 4 studies were conducted for the grow-out study for a grouper, 4 diet groups, 1 control, and 3 fed with Bacillus subtilis were comprised of 30 fish each in triplicate and were conducted for 28 days. Okay, for my bacterial isolation and identification study by Lee, 2019, the bacteria strain K2 was used in the study was isolated from the meat gut of healthy grouper. The tissue was collected aseptically and rinsed with sterilized saline 0.85% and then homogenized in 1 ml of sterile saline. The suspension was diluted 100-fold in sterile saline. Afterwards, 0.1 ml of the suspension was aseptically placed on Luria Botany agar plates and incubated at 37 degrees Celsius for 24 hours. The distinctly single colonies were randomly selected and streaked on LB agar plates. The sample process was repeated in order to get pure culture. Okay, for real-time quantitative PCR assay, total RNA was extracted from hybrid grouper distal intestine using Triazole reagent, followed by quality measurement on 1% agarose gel and yield determination with a nanodrop 2000. The RNA was treated with RNA-free DNase to remove DNA contamination. First-strand cDNA synthesis was performed using this prime script RT-PCR kit. Reaction conditions were as recommended by the manufacturer. For sampling and assessment of growth performance, the fish is collected after the experiment to be weighed and to measure the length. For the challenge test, as recommended by Lee 2019, the preliminary test was conducted before the challenge test. Vibrio harvey was grown in LB broth and incubated at 37 degrees Celsius for 24 hours. The Vibrio harvey culture was diluted to obtain a concentration of 1 to the power of 5, 1 to the power of 6, 1 to the power of 7, 1 to the power of 8, and 1 to the power of 9 cell per mil bacterial suspension. Hybrid grouper were intraperitoneally injected with 0.1 ml Vibrio harvey suspension with different concentration method above. For the result, Epinephrus fuscoguttatus study by Yasin 2016 proved that bacillus species can enhance the health state and disease immunity of tiger grouper Epinephrus fuscoguttatus. The addition of probiotic in the diet can increase the absorption of nutrient in the body of the fish. The same thing is expressed that the presence of probiotic in the digestive tract can improve microbial balance in digestive tract, thus increasing the absorption of feed and reducing the number of pathogens in the gastrointestinal tract. Claim the presence of probiotic in the digestive tract can increase the synthesis of vitamin, are able to maximize the activity of digestive enzyme in the digestive tract. For Epinephrus fuscoguttatus, hybrid with Epinephrus lanceratus, study by Lee 2016, found that bacillus species can enhance the health state and improve disease resistance against vibrosis. The increment of IgM can be seen in 28 days of feeding trial. IgM acts as first line of defense of the fish. Fish mortality was recorded daily for 1 week after challenge with Vibrio havaei in order to evaluate the protection effect of Bacillus valenciensis K2 on grouper against bacterial infection. Grouper fed with regular diet or a diet containing Bacillus valenciensis K2 showed 25% and 55% survival, respectively, and statistic analysis demonstrated strain K2 significantly increased fish survival. For Epinephilus coides, research by Pan 2013 showed that Bacillus species have the ability to enhance orange spotted grouper Epinephilus coides disease resistant against viruses. After 30 days, disease resistant against Vibrio vulnificus infection and expression of immune-related genes were determined. The study found that oral administration of B-coagulant significantly enhanced expression of several immune-related genes, including myeloid differentiation factor, MYD88, interleukin, IL-1 beta, tumor necrosis factor TNF2, and IL-1B in grouper. Okay, for my discussion, the efficiency of Bacillus species were proved and has been tested on Epinephilus fuscoguttatus, Epinephilus coides, and Epinephilus brunis. Significant decrease of FCR was observed in fish fed diets supplemented with Bacillus primulus and Bacillus clausii for 60 days. Bacillus species promotes the growth and enhance the viability of beneficial lactic acid bacteria in the intestinal tract of humans and animals. Bacillus subtilis possess anti-tumor and help in immunomodulatory activities. Dominant gut Bacillus species strain with antagonistic ability from the fast-growing fish were positively related to the growth and health of hosts. Then the last one, Bacillus subtilis A20 colonized the intestine of grouper Epinephilus coides feed. Bacillus subtilis supplemented diets will improve the feed efficiency and growth rate and induce upregulation of innate cellular and humoral immune response together. As for conclusion, Bacillus species have the ability to enhance the health state and immune response of Epinephilus species against viruses. The properties of gram-positive Bacillus species have the solution to the challenges faced by aquaculture sector. This genus have the ability of rapid growth rate, tolerates a huge range of conditions, and the ability to sporulate. Bacillus species possess anti-tumor and helps in immunotherapy activities, thus promote the growth and enhance the viability of beneficial lactic acid bacteria in the intestinal tract of Epinephilus species. That's all from me. Thank you.